The overall goal of this large-scale electron microscopy, or nanotomy experiment, is to define alterations from the macromolecular level to the tissue level, in today's case in the degenerating zebrafish brain.
Nanotomy can help answer key questions in life science, for example, in neurodegeneration and in type 1 diabetes research.
The main advantage of nanotomy is that unbiased information is acquired, ranging from macromolecules, organelles, cells to tissues. This allows quantitation and open access data sharing via nanotomy.org.
Although nanotomy provides insight into immune maintenance and microglia in the zebrafish neurodegenerative brain, it also is applied to other disease models, including cell culture, mouse and even human tissues.
Generally, scientists new to this technique struggle because the amount of data is overwhelming. This can be 1,000 times more than obtained with traditional EM.
After fixing zebrafish larvae according to the text protocol, cut the larval heads rostrally to the hindbrain to facilitate penetration of osmium. Place the larvae in 1% osmium tetroxide, 1.5% potassium ferrocyanide in 0.1 molar sodium cacodilate and incubate on ice for 2 hours. Then use double distilled water to rinse the embryos 3 times for 5 minutes each time. Prior to embedding, the samples need to be dehydrated in a series of ethanol. To embed the embryos, after incubating in diluted epoxy resin overnight according to the text protocol, remove the diluted resin and use pure resin to replace it. Incubate for 30 minutes, then replace the resin a second time and incubate for another.